In this session, we will study the simple and conjugated proteins with some examples, and also we will understand the primary structure of proteins. What is primary structure of proteins? Now, simple proteins. We already know that amino acids are the basic units of the proteins, and the proteins which are made up of only amino acids. If the protein molecule is made up of only amino acids and no other component like carbohydrate or phosphate group, etc., then such proteins having only amino acids are called simple proteins because on hydrolysis they yield only amino acids. If these proteins are broken down by the action of enzymes, only amino acids are yielded. And example for simple proteins are albumins, which are soluble in water, and globulins, which are insoluble in water. We know that albumins they are very important in nutrition as a source of food, and globulins have different functions like immunity, etc. So these are simple proteins. And other simple proteins, example for other simple proteins are glutenins. They are soluble in dilute acids and alkali but insoluble in water. And prolamines, soluble in 70% alcohols. Histones, soluble in water and very dilute acids. Protamines, simplest of proteins. So, glutenins are found in some plant products, and histones are part of the DNA and RNA. I mean, they are part of the chromosome. Okay. So, like this, there are many proteins which are simple, made up of only amino acids. Now, next one is conjugated proteins. As the name suggests, these are the proteins which are composed of proteins and non-protein substance. So here what happens, a non-protein molecule is bound to the protein molecule, and such structure results in the formation of a conjugated protein. So the non-protein component is called prosthetic group or cofactor. Now, prosthetic groups or cofactors are small organic molecules which are normally formed from the vitamins. And on hydrolysis, they yield amino acids and also the other chemical components which are nothing but the prosthetic groups. Now, examples for conjugated proteins include nucleoproteins, glycoproteins, phosphoproteins, and hemoglobins. Now, here nucleoproteins, so as the name suggests, the proteins which are associated with nucleic acids like DNA and RNA. And nucleoproteins are nothing but chromosomes, right? Then chromatin material of cell is a nuclear protein. Now chromatin material, I mean chromosomes. Then glycoproteins means the protein molecule is associated with carbohydrate. Then phosphoproteins, where the phosphate groups are attached with the protein molecule. Hemoglobin, globin protein which is associated with heme, that is Fe. And hemoglobin, it is a very important pigment which is associated with transportation of oxygen in the blood. Then glycoproteins, as I already mentioned, they are carbohydrates which are associated with protein. Hyaluronic acid, chondroitin sulfate, and heparin are example of glycoproteins. Then phosphoproteins, we studied chromoproteins, lipoproteins. When proteins are associated with lipids, they are called lipoproteins. Now there are some metalloproteins. When the protein molecule is associated with metal ions, they are called metalloproteins. So the protein molecules may be associated with iron, zinc, copper, etc. So most of the pigments like the chlorophyll, hemoglobin, chlorochlorine, etc., they are all example for metalloproteins. Now metalloproteins are commonly called pigments, and they have many functions, and one of the important function is they are responsible for coloration. For example, hemoglobin is responsible for the red color of the blood. Then the coloration of the plants, the flower colors, etc., leaf color, green leaf color, etc., they are mainly due to the presence of such pigments, and these pigments are nothing but metalloproteins. Now let us move on to the study of primary structure of proteins. Now, understanding protein structure is very important because proteins are involved in almost all biochemical reactions of the body, and the functioning of the protein is associated with its structure. Now, this is what you have to remember. So, the shape of the protein is directly responsible for the functioning of the protein. If there is any change in the shape, then it becomes non-functional, right? So therefore, understanding the primary structure, secondary structure, tertiary structure, and the quaternary structure of protein becomes very important. Now let us start understanding the structure of protein by studying the primary structure of the protein. Now you know that proteins are very large molecules which are made up of amino acids. Now what happens? There are 20 different types of amino acids. So amino acids are bound with each other by means of which bond? Peptide bonds, right? So one amino acid and another amino acids are bound with each other by means of very strong peptide bonds. Of course, these bonds can be broken down by peptidases, the protein digesting enzymes. Now, a chain of such amino acid. Initially, what is a protein molecule? Protein molecule is a chain of amino acids. So the chain may be like 20 amino acid long, 200 amino acid long, 20,000 amino acid long. It depends upon the type of protein. So you find a sequence, okay, of amino acids. So for example, the first amino acid is leucine. Second amino acid is histidine. Third amino acid is isoleucine. Fourth amino acid is 309, like this. So if you write down the name of the amino acids, so you find a sequence. So this sequence of amino acids is considered as the primary structure of the protein. Now this is what you have to understand. The sequence of amino acids in a protein molecule is considered as the primary structure of a protein. Now, why it is important? You know, there are infinite number of protein molecules that exist in the nature. Even in our body, there are thousands of different types of protein molecules. But every protein molecule is made up of same 20 amino acids. Now take example. Insulin is a protein. It is a hormone molecule. Okay, and another protein you can take like melanin. Now melanin is also a protein, but melanin and insulin. Melanin is a pigment, insulin is a hormone. They have two completely different functions. Insulin is involved in glucose homeostasis, whereas melanin is a pigment which is responsible for coloration of the skin. But chemically, melanin and insulin are made up of amino acids. If you break down, you will get the same 20 amino acids from melanin and same 20 amino acids from the insulin. Then what is the difference? The difference lies in the primary structure. The same 20 amino acids are arranged in different sequence in melanin and insulin. Say for example, you take the English letters, right? The same letter, for example, uh, now for example, take a word like rail, R-A-I-L. So you know what is a rail? The meaning, and it is made up of 4 alphabets: R, A, I, L. So you can think of R, A, I, L as amino acids. Now you change the sequence. You use the same 4 letters like this: L-I-A-R. What happens? It becomes liar. So RAIL, LIAR, both are made up of same letters, but they have two different meanings. Liar is a different word, rail is a different word. So this is how the same amino acids will create different protein molecules by changing their sequence. Now just think of the permutation combination with 20 different amino acids which can combine in different numbers. So that's why with 20 simple letters like amino acids, there is infinite possibility of proteins in the nature. So therefore, the primary sequence of amino acids is considered as very important, and that is the primary structure of proteins. Now, who decides this primary structure? So who will tell, or who is responsible for this arrangement of first isoleucine, second histidine, third proline, fourth isoleucine, like this? It is the DNA. It is the gene. So the information for this linear arrangement of amino acids is actually situated in the gene. So during the protein synthesis, that sequence is produced. So gene is nothing but the information in the gene is nothing but the information of the amino acid sequences, nothing else. And why this primary structure is so important? Because primary structure of the amino acids, a primary structure of the protein decides the secondary structure and tertiary structure and quaternary structure. If there is any change in the primary structure, there will be a change in the secondary structure and also the tertiary structure. Now you remember, tertiary structure or the three-dimensional structure of the protein is responsible for the functioning of the protein. So amino acids, you know, they have different properties like hydrophilic, hydrophobic, acidic, basic, like that. Different proteins have different properties. That's why the arrangement will lead to a definite uh, secondary and tertiary structure. So that's about the primary structure of proteins, which is very important. So what is primary structure? The sequence of amino acid arrangement in the protein is called the primary structure of proteins.